title of the article, the title, abstract, keywords. Selection of the keywords. So, introduction, methodology, results, discussion, conclusion, references. So, these are the essential components of the research article. Now, when we talk about the title and the abstract of a research article is like the face of the paper. It's like face is the index of the mind. Similarly, this should be attractive. The title and the abstract should be attractive. Why? Because it is the gist of the paper which will allow the reader to further go ahead and read your paper. So it should be attractive. The title should be clear, descriptive. It should also show what the trial, what the study design you have selected, what the title is, carefully selected keywords within the title to improve the article's discoverability. So from the database, when you are going to search, for example, if you are searching through a literature review or whether it is a clinical review, you want to study about the curcumin, healthy. So directly if the keyword is healthy and if you are wanting to find out about the curcumin, that should be the keyword so that it will ensure that the reader will type and they will get the paper. So it is very important to make the title attractive. Again, there will be a series of lectures which will tell you how the title has to be framed, what is abstract, what are the keywords. Again, in the further sessions, the detailed explanation will be given regarding this. Right now, I will be only talking about animal research. First, we are talking about experimental research. In experimental research, these are the essential components. Further, you will be studying about the title, studying about the keywords and abstract. This is just a brief introduction. So, how your abstract should be? An abstract should be structured abstract. So, what does it include? It should include the introduction with the background of whatever topic you have selected. Apart from that, it should include the methods. What is the methodology you have adopted? What was the trial? Whether it was an for example, open observational study, or whether it is a randomized controlled trial. What the methodology you have selected for the trial has to be mentioned there with the key findings. What are the interpretation of your research finding? Even that should be written. Then conclusion. Conclusion of your trial should be mentioned. So the abstract will give you the total information of your trial only if it is attractive and if the reader find it's of their interest, then only they will further go and read your complete paper. So the keywords, again for searching in the databases, select the proper keywords because it will be very helpful to find out what relevant topic they want to search. Next slide. So introduction, methodology, statistical analysis. What is introduction? Though this is the first part, but it should be written at the end. The introduction after completion of your research, introduction should be given. It should have some background information. What are the previous studies? Why have you selected this trial? What is the research question? Then it also should clearly state the specific objectives. Why have you taken up this? Why do you want to conduct this particular clinical trial? What are your objectives? And what are your expected outcomes? Even that has to be included in the introduction. So methodology. Methodology is a section wherein you will write down about the design, study design. You will write down about everything in methodology. The inclusion and exclusion criteria, how you are going to select your subjects, what is randomization, everything will be mentioned in the methodology section. How are you going to collect your data? For example, in the clinical trial, history taking, laboratory investigations, examination. So all the details will be mentioned in methodology. For example, the experimental workout, what investigations, what are the biochemical markers, and how will you just collect the data. Everything will be given. For example, if there are two groups, so you are going to write two experimental groups. Now since we will focus on the animal trial, if there is a test group, you are going to write about the test group and the control group. If you are taking comparative trial, then the comparative trial you will write group A, group B. It will not be test and control. And in case of a comparative trial, it will be, the ratio will be equal, 1:1 ratio, the sample size estimation. And in case if you want to do on a standard control and the test drug, then you can take the ratio as 2:1 or 3:1. So again, sample size estimation, you can always go to the biostatistician and tell about what your intentions, what the protocol should contain. When already standard drug, whatever standard drug you have selected, already it is proved. Then again, why to give more sample size to that group? Instead of that, the test group, you can increase the number of the ratio, 2:1 or 3:1. So based on your protocol, based on your understanding what you want, you have to discuss with the biostatistician, and the sample size has to be estimated. Even that has to be given in detail in the methodology section. The inclusion criteria, the exclusion criteria, your outcomes, everything will be mentioned in detail. Then statistical analysis. What is this analysis? After completion of the trial, what are the results? How you are going to analyze it? What are the tests which you have used for the analysis? That will be mentioned in this section. That is called as statistical analysis. Next. Now the results, discussion and conclusion. Again, result section, using tabulated diagrams, figures, you are going to give your results in detail, in an organized way. Then again, discussion. This part I will leave because again in the next coming session, they will in detail explain you what is discussion. And again, interpretation, sample size, methodology, everything will be dealt in detail. Conclusion. It has to summarize the key findings of your trial and what were the limitations and what are the future recommendations should also be written in the conclusion. Next. Now comes, what is this experimental research? The experimental research can either be a human research or an animal research. If we are employing human subjects, then it is called as a human research. If we are employing non-human models or the animal models, then it is called as animal research. So today my focus is on animal research. See, the human trial, whenever a human trial is being conducted, it is necessary to actually get an institutional ethical committee for biomedical research approval and institutional scientific review board. There will be a scientific review board. First you have to present your protocol, which is called as the synopsis, to the review board. Once it is accepted, again institutional ethical committee. So only after getting this clearance, then you can start with the trial. It is only for the protection of the human rights, and so it is very important to take the informed consent. Whereas in animal studies, we have to take institutional animal ethics committee approval before proceeding for the clinical, whatever the trial. Then both types of research are held to high ethical standards to safeguard the well-being of the participants, whether human or animal. Only for the safety, their protection, the ethical issues covered, the patient's consent, information to the patient in humans. And otherwise, even for the animal safety also, it is very important to take the approval. Whether it is a human trial or whether it is an animal trial, ethical approval is important. Now the human trials. Here in human trials, it involves the direct interventions. It is usually a safety trial. For example, a randomized controlled trials, blinding, placebos. Either you can go for RCT with a placebo and a test drug or standard drug, or comparative trial, randomized comparative, where you are going to test two drugs at a time in two groups with equal number of sample size. In case if you are going for standard control, the drug which has been FDA approved, in that case, you will keep a standard and you are tested, whether it is Unani, Ayurveda, whatever particular herbal concoction or any powder, Kashaya, Joshana, whatever. The thing you have to mention, it becomes a randomized comparative clinical study. If you are comparing two drugs, if you are comparing to the standard, it becomes randomized standard controlled trial. If you are having a placebo in one particular group, nowadays it becomes an ethical issue to use only placebo. And placebo, it will not be allowed. But sometimes we have to prove and we have to take placebo. At that time when you are taking placebo, you should also tell about the rescue medications you are going to use. For example, in heavy menstrual bleeding, we cannot go for placebo. If at all we are going for the placebo, rescue medications, what rescue medications you are going to give, even that has to be mentioned, and small number of sample size will be taken for the placebos. And in case if the patient does not respond, then immediately we have to switch on to other therapy. That should be mentioned. Now, animal studies utilize experimental procedures on non-human animals. See, these animal models can be albino rats, can be guinea pigs, whatever the albino, whatever the non-human, that is models, the animal models. We can do surgeries, we can administration of substances, intramuscular, IV, whatever we are doing, genetic modifications, everything has to be mentioned in detail. Then the complementary approaches. Why the methodologies differ? But the findings from both human and animal research work together. Why? To have an advanced scientific understanding and also to improve the healthcare system. So that is our main motto. Next. See, what are the outcomes and applications? In the human trials, it directly evaluates the safety and efficacy, the clinical outcomes of the interventions for potential human use. Whereas in animal studies, these are a preliminary insights into biological mechanism and responses informing the development of human trials. So these animal studies, they should help in replication or replicability or reproducibility. What do you mean by reproducibility? They should form as a basis for further future researches. In animal trial, US has spent billions of dollars on animal trials and 50% of them go waste. Why? 50% of them go waste because they have not selected a proper design. At the end, the result, either sometimes it will be negative or sometimes it will be inconclusive. If it is negative, it still holds good in the research. Even negative results have some important role. We'll come to know that this particular medicine what we have used has negative result. But if the results are inconclusive, you have planned a protocol, you have hired so many things, you have gone with the biochemical markers, investigations, animal setup, everything, and finally the results are inconclusive. Why? At that time, when the results are inconclusive, you have to sit and plan and change the study design. So they are spending millions and dollars on animal research so that if they are spending so much, they should also be cautious. And this type of preliminary insights on animal research, if they are published, then it will be very helpful in reproducibility in future. They will take this as a foundation and again continue future research. So therefore, we will focus on animal studies, how to do the research and how to publish an article based on certain set of guidelines that we will be discussing later. Now, what is the impact of both? The impact of the integration or the impact of both human and animal research findings accelerates the translation of scientific discoveries, which will be helpful, and the healthcare system will also improve. So now comes the AYURVEDIC guidelines. These AYURVEDIC guidelines are nothing but a set of guidelines recommended for animal research reporting. If you want to write the article on animal research, these are the guidelines which you are going to follow. And before that, since you want to publish a paper, and if you don't follow the guidelines, the paper will be rejected. So actually you should start planning your protocol based on the AYURVEDIC guidelines. What the guidelines require, you can't do anything once the protocol has been accepted or approved by university. Your ethical board has approved, your time when you come to know that your paper has to be published, and especially the animal experimental trial has to be published using the ARIVE guidelines, at that time you will find out that so many things you have missed out in the protocol. So it becomes difficult for you to publish. It will be rejected. So it is always better, while planning the protocol, first read the guidelines and read what the specifications are required and which are very important, without which the paper cannot be published in future. So therefore, what are these ARIVE guidelines? The ARIVE, Animal Research Reporting of In Vivo Experiments. See, the guidelines are a set of recommendations that aim to improve the reporting of research using animals, enhancing the transparency and enabling others to reproduce the findings. So, replication, reproduction of the trial in future and transparency has to be maintained so that it will be very helpful to go for further future researches. This presentation will provide a comprehensive overview of the ARIVE Guidelines 2.0. These ARIVE Guidelines 2.0 is a revised guidelines. Though this scientifically it has been by the scientific community, these guidelines have been accepted but not being followed. There is some problem. With ARIVE Guidelines 1.0. Therefore, they have revised the guidelines. These guidelines are called as ARIVE Guidelines 2.0, equipping researchers with the knowledge to effectively implement these standards in their animal studies. So what are these ARIVE Guidelines? I told you, they are revised and they contain a set of 21 items that provide a framework, a structure of your trial for reporting animal research in a comprehensive, transparent and standardized manner. Why are they important? These ARIVE guidelines help to improve the quality of animal research, enhance reproducibility and increase public trust in the scientific process. With all details mentioned, you are going to mention about each and everything in the paper. What the study design, how you have taken care of the animals, what feed was given, what is the inclusion and exclusion criteria, what are the biochemical markers, everything in detail will be mentioned. The ethical approval, the clinical trial registry, everything in proper sequence as per the guidelines will be mentioned. So therefore what happens? There will be more trust, the public trust, and also it helps in transparency. So evolution, I told you, the evolution of ARIVE from 1.0 to 2.0. The 2.0 is the latest one, the revised one, and this has to be followed. Now the experimental groups. Clear, see, these ARIVE guidelines focus on the study design. For example, if you are including experimental groups, clearly describe about the experimental groups. If you are having 2 groups, if you are having 3 groups, please mention group A, group B, group C. Or if you are having test group, mention test group, then control group. And please mention what is the medications given, the complete history of all the medications, whatever you are using. So experimental groups, you are going to enter in detail about the experimental groups, whether test group or whether it is standard. Interventions. Provide a detailed description of the interventions being used, including the dosage, the frequency, the route of administration, whether you are giving per rectal, whether giving intravenous, it has to be mentioned. Whether you are giving how many times, frequency, the dosage, everything in detail. Then the outcome measures. Again, clearly define the primary and the secondary outcome measures. What are your primary outcomes? That is the main outcome of your trial. So the primary and the secondary outcome, if you have used a certain scale, for example, any pain scale, even that has to be mentioned. That will be assessed, including any relevant behavioral, physiological or biochemical parameters. I told you biochemical markers will be used sometimes to know what was earlier, because in experimental models we will they are also called as induced models, wherein one particular animal model, say albino rats. We will inject, we will produce, actually there will be healthy models. These healthy models, we will inject and cause the disease. After injecting and causing the disease, you will give your medications. Before that, you will take all the investigations, what are the biochemical markers, then again produce the disease, again give the medication, then again repeat the biochemical parameters, so that before and after everything will be present in detail. Sample size determination. So describe the methods for calculating the appropriate sample size. See, based on your previous study, how will you calculate a sample size? Based on a previous study, I told you, reproducibility. Based on a previous study, you will just write down what is the previous study you have taken for your inference, so that it will help you to calculate the sample size with the power analysis, with 80% of power, 90% of power, what is the error, everything will be mentioned. And again, this will be help, the biostatistician will help you. Only you have to tell about your ideas. In case if you are wrong, they will help you. They will help you to find out the paper also. You want to calculate the sample size as per your convenience. If you are not able to calculate with the previous study, you can use one scale. For example, paid scale. You can use, for example, 2:1 ratio. If I am not able to get the sample size with the previous trial paper, I will take one scale. But either hook or crook, I will try to make the sample size as I want. But that can be done by a biostatistician. While you are doing the synopsis, you should tell your ideas. What are your ideas? And they will help you in a proper way to exactly find out the sample size. Now, justification for the sample size. After selecting that particular 2:1 ratio, 3:1, the same ratio, previous study or a scale or a score, whatever you have selected, you have to justify why you have taken up this. Because of XY, you should say standard and test group, you would like to go for a 2:1 ratio. If it is just a comparative, you have taken two drugs and you have to compare, then you have to take equal number of samples. Just give some justification. Even the justification will be given by the biostatistician. Then adherence to the ARIDE. Reporting the sample size and its justification in accordance with the ARIDE guidelines enhances the transparency and validity of the study's findings. So always, while framing the protocol, it is called as the synopsis writing, always first keep the guidelines and then frame. Because in future, you will not have any issues in publishing your article. Now, for example, this is the sample size. How you will write down? The sample size calculation was based on post-operative paid numerical rating scale. So, based on the scale score, the sample size was calculated. Then there is a software which experimental, I have shared the link also. With that, and we'll also say the power of the experiment was set to 80%. A total of 20 dogs per group were considered necessary. So there will be two groups. In each group, 20 animal models. Here the animal model is dog. So you will write 20 dogs in each group. Next. Inclusion and exclusion criteria. It is very, very important. Inclusion and exclusion criteria. Which animal models you are including in the trial and which animal models you will exclude from the trial, it has to be given. For example, you can just see the example. In a study on a new drug for arthritis, the inclusion criteria may specify adult male rats 12 to 18 months old. If they are fixing in that range, 12 to 18 months old, then only we will include in the trial. With a body weight between 300 to 400 grams. With a body weight of 300 to 400 grams and no pre-existing health conditions. So these are the inclusion criteria. For example, if the weight is so and so, then only I am going to include. Then we will write if there is any systemic illness, then we will exclude. If there is any pre-existing health condition, then we will exclude from the trial. So these inclusion and exclusion criteria are very, very important. See, factors like age, weight, sex, genetic background, health status, everything to be mentioned in detail. Next slide. A hypothetical study on the efficacy of a particularly rats. What will be my inclusion criteria? The male and female rats aged 2 years exhibiting early signs of heart disease, in whom early signs have already started. Then exclusion criteria: rats with severe kidney dysfunction or those previously treated with beta blockers if they had hypertension or if they have any kidney dysfunction. We will exclude those animal models. Why? We are actually testing for cardiac medication. For that, safety and efficacy both has to be calculated. For safety, we will do a liver function and also renal function. Already if the animal model has some problem, then definitely we have to exclude. But we will mention in our exclusion criteria, because while publishing, this is very important. You have to mention the rats with severe kidney dysfunction or those previously treated with beta blockers will be excluded from the study. So what is the impact? How this inclusion and exclusion criteria will help you? Because it will help in the uniformity. There will be no bias, no selection bias, and also the uniformity or homogeneity in the study group. It leads to more interpretable results. Exclusion of certain condition prevents confounding factors, some kind of confusion. That confounding factors, if they have to be prevented, you have to exclude certain conditions and enhances the safety and ethical compliance of the study. So therefore, exclusion of certain conditions where we think it is necessary, you have to exclude. Next, what is randomization? See, randomization is the process of randomly assigning a subject to either of the groups. You are not why randomization has to be done? No selection bias. Why no selection bias? Maybe you are thinking whatever drug, there is some hypothesis in your mind that maybe group A will one test group, one control group. So you may be thinking one group will have better effect. So you will try to more focus. Unintentionally you will be focusing. Therefore, selection bias to be avoided. How it can be avoided? By giving randomization. So either computer generated randomization, it will help in avoiding selection bias. So what is the, I told you, selection bias can be avoided, so that is why randomization will be done. Now researchers can use the random number generators and say computer generated randomization number, or there is a software to assign this animal model to either the control group or the treatment group. Next. So no, please. Next. Blinding. So what is blinding? See, blinding is a process wherein the researchers or the participants, they will not have the knowledge what medicine they are being given. So in case of animal models, the animal caretakers, the assessors, and also the research participant, they will not know. It can be a single blinding, it can be a double blinding, or it can be a triple blinding. So in a study testing a new pain medication, the investigator administrating the treatment and the animal caretakers are both blinded to the group assignments. So if only one caretaker is blinded, it is called a single blind. If the assessor as well as the research who gives the medicines, all the three are blinded, it becomes triple blind. So that is called as blinding. Next. Next. Okay, outcome measures. So, what are the outcome measures? What was the outcome of your study? That has to be, since we are using animal models as per the guidelines, the outcome measures based on the survival rate. How many animal models were included? At the end of the trial, how many survived? How many died? The survival rate. The survival rate has to be mentioned. Then the behavioral changes. What were the behavioral changes in the animal models? Whether they were stressed. There are some, like we will put a simple kind of a test wherein the rat will be allowed to swim. We will see how the rat will swim or the animal model. So based on that, the anxiety level, the stress level, everything can be calculated. These are called as the behavioral changes. Then the physiological measures. What are the physiological measures? All the I told you blood investigations. What were the earlier investigations? After the treatment, what are the changes in the biochemical markers? Even that has to be tabulated as an outcome measure. Histological analysis. This involves examination, that is the microscopic examination. So therefore, for example, if you are doing a research on tumor cells. So what were the tumor cells? What were the number? What was the severity? After giving your medications, what is the number of the, like, is the tumor reduced? That histological analysis should also be, depending on what trial you have taken, histological analysis should also be considered as an outcome measure and tabulated. In a tabulated form, it has to be documented. Then the biochemical markers. I told you hormones, anything, blood, urine investigations, the tissue culture, anything, it can be considered as the biochemical markers. Even that has to be documented, pre and post. Now the statistical methods. They provide details of the statistical methods used for each analysis, including the software used. What type of test you have used to analyze the result, that has to be, whether it is ANOVA, whatever the test they have. Again, the biostatistician, after you have completed your trial, you are going to enter it and send it to the biostatistician, and they will tell you what kind of test they have employed to give the results. That is called as the statistical analysis. And what were your assumptions, and whether you were able to meet the criteria, whether what was your hypothesis, and what exactly the outcome is. Everything has to be documented. Next. Now the experimental animals, the species and the state. What type of species, what are your animal models, everything should be given in detail about the animal model. Their sex, age and source. From which source you have procured the animal models, even that has to be mentioned. Additional characteristics. Provide relevant details about the animals such as their health status, weight, pre-existing conditions. I told you inclusion and exclusion criteria, all the things will be mentioned. Again, about the experimental animals, all complete details have to be mentioned. Now, report the correct nomenclature. It is very crucial to understand the data and ensuring that the research is discoverable and replicable. So see, for example, if you are collecting mice, what is the standard nomenclature? I have even used the website address where you can click and just read about the details. What nomenclature has to be given. For example, if you are using rats, zebrafish, Drosophila, whatever the animal model is, the particular nomenclature is given, and everything is given in the arrival guidelines which I have already shared with the coordinator. Next. Now the experimental procedures. What are the procedures? It has to be given in detail. See, step by step, the description of the experimental procedures, whether you have included any modifications, what was your protocol, and did it require any modifications, then that modification should also be entered. Reporting of the settings. What were your settings? For example, what happens? There will be a bias. Why there will be a bias? For example, there is a cage and there are racks. If the animal is placed on the top rack and the animal placed on the lower rack, the animal which is placed on the top rack, maybe because of the heat, because of the electricity, maybe there will be some kind of stress. So therefore, the setting should be uniform. Uniform setting is very, very important. So therefore, why this is very important? To maintain transparency. The outcome, the outcome will differ in case if you are not maintaining the uniformity. For example, if a surgeon is performing a surgery on the animal model, the same surgeon has to perform the surgery. If you are keeping in a certain environmental condition, the same environmental condition should be maintained for all the animals. If you are giving a particular feed, the same feed to be given. A particular time is maintained, the same time has to be maintained. So this all comes in uniformity so that it ensures a transparency. And all this is done for future replicability and reproducibility, because our main concern of this research is for future references, and so that they can take this research forward. Arrive compliance, adherence to the Arrive guidelines in reporting experimental procedure enhances the overall quality and reliability of the animal research. Then only you can carry forward and then conduct the human trials. Next. See, examples, the same thing. The examples of reporting the specific types of experimental procedures. What are the experimental procedures? Everything in detail. Next slide. Now, reporting results. Data presentation, significance of the reporting, transparency of the reporting, with following all the above guidelines, our results, it can be tabulated, and we are sure enough they are transparent. These results can be tabulated in figures, in tables, in diagrams. We can tabulate our results. Next. So again, recommended set: abstract, background, objectives, ethical statement, housing and husbandry, animal care monitoring, interpretation, the results, the conclusions, the protocol registration, what is your CTRI number, where you have registered, what is your ethical approval number, everything, declaration of interest. This is the recommended set for your paper to be published. Next. So, already I have told you the title, the 15 minutes. So, neuroprotective effects of the compound XYZ in a mouse model of Alzheimer's disease. This is the effective title. You can understand by this. XYZ is whatever medicines you are going to use, you can write it. Instead of that, animal study of a new drug for Alzheimer's. Ineffective title. That is an example. This is an effective title, and that is ineffective title. Therefore, these are the examples how you can improve. Key considerations. The title should be specific. It should be informative. It should be attractive. It should tell about the animal model. For example, in mouse, in adenorats, in guinea pigs, in dogs. That intervention and the animal model has to be written. So that becomes the key considerations for your title. The titles that meet Arrive guidelines provide clear, concise and comprehensive information about the study, enabling the readers to quickly understand the scope of the study and also the relevance. Next. Abstract. Same thing, it should be structured with background, objectives, methods, then analysis, your conclusion, finally the keywords. That is abstract. Next. Introduction. I have already told you it should have some information about the disease, the previous studies, and how you want to bridge the gap. What is your new study? Why do you think your study is important? Why do you think this study has to be done, has to be conducted? All those points have to be mentioned. Next. Same thing. I told you, the filling your knowledge gaps, what is your objective, and what is the scientific background. Everything to be mentioned in introduction. The ethical approval number. Then animal welfare measures. What step you have taken for the animal welfare? Have you taken the ethical approval? Apart from that, how did you maintain? Justification for animal use. Why do you think this particular animal model is required in this particular disease? That justification. You have a book on animal models to be used, what animal model in what disease. So taking that, you have to give the justification. Housing and what are the housing conditions? How did you maintain? What is the animal? Based on this Arrive compliances, everything, how did you maintain? That has to be written, one pointer at least. Next. Animal care and monitoring, that's same. Housing conditions, what feed you have given, how did you monitor their health, all ethical considerations, all have to be mentioned. Then the scientific implications. The primary outcomes of this animal study will measure the efficacy and safety of a novel drug candidate for treating a rare neurological disorder. So whatever you have taken, you have to just write down. What were the findings. The findings could have significant relevance to human health, potentially leading to a new treatment option for patients suffering from debilitating condition. So you have to justify why did you take and what is your hypothesis, what could happen in future, everything. Next. Then species relevance, external validity, the factors influencing the generalizability. So what are these factors? How can you have the limitations? Include the controlled laboratory environment. You have to tell about the limitations because you are conducting this in a controlled environment. What will happen if you are going for further trials, phase 2, phase 1 trials? So therefore some limitations, whatever you feel that limitations should also be mentioned. And also you have taken a small sample size. You should write about the limitation. This particular trial was conducted on a small sample size. So future recommendation, the same trial to be carried on a large sample size. Next. So these are the examples. Protocol registration. I told you first thing is you have to register your protocol. Get the approval, then start with the trial. And you also should mention the time period of the trial. You are going to complete this trial within 1 year, within 1.5 years, whatever the time period should also be mentioned. Next. Then the data access and sharing. For example, if any particular person after reading your paper gets back to you, then you should tell that we are committed to sharing our research findings with the broader scientific community. So some findings, if anyone reads your paper and come back to you, then before that only you should tell, we will give access to the data for reasonable request. For example, if you want to conduct future research based on our research, then we will definitely provide the data. Next. So declaration of interest. There are no conflicts of interest for the researchers. At the last, you have to write down this sentence as declaration of interest and also who has funded for this research. And you have to acknowledge the authors and whoever have contributed for the study, for the trial. You have to write down. Next. Then you will come to the conclusion. Summary, implications, what are your future recommendations? Next step. Proposals for future research directions. How this could have been in a better way. What practical difficulties we faced and what do you recommend out of your research? What did you learn? What do you recommend? That has to be done. Next. Now this is the group activity. How many more minutes? 10 minutes. So, see, you should open the RF guidelines which I have shared and then next slide. Testing anti-inflammatory effects of a herbal extract in a rat model of asthma. This will be the title. If this is the title, next slide. What will be our objective? The objective, only the title will be given. The rest all you have to fill. So how? To investigate the anti-inflammatory effects of a new herbal extract on airway inflammation in rats induced with asthma. That will be our objective. To see what are the anti-inflammatory effects of a herbal concoction. Then study design. Double-blind, placebo-controlled trial involving again, this is all you have to think and just write. If you want for a triple blinding, then if you want a single blind, you can just think of it and write down. What will be your control? What will be your placebo? What is the herbal extract? What is the exact formulation that can be written? Then ethical review. Then you should say institutional ethical review has been approved. The institutional ethical for the biomedical or the animal research, we have the approval and you should write down the number also. Ethical approval number. Then the sample says justification. What is the previous study you have taken? Okay, based on what did you calculate the sample size? How you will justify the justification as a preliminary data you have to write. Then the animal details. I told you, you are selecting a rat model. Then you will write down the rat model. Whether you want to take only the female rats or the male rats or the baseline health status. Everything Housing and husbandry describes the controlled environment settings at what temperature it will be maintained, humidity and housing conditions. Everything will be mentioned. Next. So, what is your experimental procedure? What is the dose of the drug? What is the method of route of administration? Then outcome measures. Based on what will you go for your outcome measures? Lung inflammation markers. Secondary outcome, overall lung function and physical activity levels. So always in an outcome there will be primary and secondary. So you will write down lung inflammation markers, the biochemical markers, which will help you, and they are the main outcome of your study. Whichever is the main outcome, that becomes primary outcome. And for example, if your overall assessment of the health, overall assessment of the lung function, that becomes your secondary outcome. What statistical methods you have used to analyze your results, that statistical methods you have to write. The results and interpretation, it has to be given in a tabulated forms, figures, bar, charts, all those things, bar diagrams. Then funding and conflict of interest. You have to write down who has funded for this particular research and there is no conflict of interest in the study. These are the references. Thank you. Again, next. Immediately. No, activity I just told you. 5 minutes.
Delegates are requested to be seated. We will continue.
So my second lecture is on drafting drug research-based original article. Earlier it was experimental research and we focused only on animal research. Now we will focus on drug research. Like animal research, we had certain set of guidelines called as RI guidelines. For drug research, there is no particular set of guidelines. No guidelines. Therefore, it becomes easy. Like all the other literary review articles, in further sessions, you will be learning how to write a research article based on a set of guidelines in case of a RCT, in case of a literary review. If you are using a meta-analysis study, what are the guidelines? We have a set of guidelines for everything. But for drug-based research, we don't have any particular guidelines. But there are certain important points which you have to focus on. See, what are the objectives? The participants should be able to understand a basic concept of writing drug research-based original article. So only on the particular drug. To develop the skills for proper summarization of the drug research-based work. To plan a research work for drafting good articles based on drug or medicinal formulation. It can be a single drug or it can be a formulation. Scientific explanation of its therapeutic actions, exploration of its various uses with the safety and efficacy parameters. So at the end, you should know at the end of the session what is drug-based research and how it has to be written, how the article has to be written. Introduction. See, the drug or the formulations are an important tool for obtaining ultimate goal of treatment. See, the quality of medicine is achieved by continuous research and development. So therefore, the propagation of knowledge of the ongoing research and development process on a particular drug is the key of improvement in the medical research field. Even if you know that this particular drug has effect on a particular disease, you can continuously do this research to minimize the drug, the dosage form. For example, we are using the crude in the form of powder, in the form of saffron. If we are using 7 grams of powder 3 times daily, you know, if you make 7 grams of powder into tablet form or in a capsule form, this number of tablets, this number of capsules you will have, ask the patient to consume. Only apart from infertility, they will not agree. Only in infertility, because of so much of pressure from the family, from the mother-in-law, from the husband, so they consume 7 grams or 10 grams also they are ready. But for any other disease, if you give this amount of medicine, they will simply nod their head daily, but definitely they will not eat. They will not consume the medicines. So it is better, whatever it is written in classical books, we can go for the new dosage forms. We can go for dosage forms. First, we will find out. Later, we will just see the dosage forms with a different dose. What is the action? Initially, we will take powder. In the powder, the patient compliance was not good. We can write the patient compliance was not good. Therefore, there was maybe acidity. The patient was not, comfortable taking so many medicines. So we will try to give the extract. We will try to give the extract of the same drug. Then again, in the future research, we will try to find out the nanoparticles of this particular 7 grams only. In the 7 gram dose, what was mentioned in our classical literature. Now, in the ethical approval, they ask, what is your reference? From which book did you bring this formulation? What is the dose given in the book? They ask, right, in the ethical. Therefore, you can take 7 grams only. That 7 grams, you have to reduce it to nanoparticles and see what is the dose in the nano amount and give for the patient and see what is the effect. The dosage form can be changed. So therefore, this is a continuous process. Proper way of planning of drug research as well as drafting of its original article is an important step. Next. See, what are the points to be addressed in drug-based research article? If there are guidelines, whenever you want, if you remember the guidelines, you can just take the guidelines, sit and study the guidelines and plan the protocol. Or you have already planned the protocol, you want to publish an article based on the guidelines of the journal. Again, the journal has a separate set of guidelines. Based on the journal guidelines also, you can sit and write the paper. But for this drug research, there are certain points which needs to be addressed. Points to be included in the article. Standardization and validation, drug identification and authentication. After drug identification, from where you got it identified, who has authenticated, even that letter has to be attached in your thesis or attached in your paper. Safety and efficacy of the drugs, how it was proved, what are the parameters. Again, we will discuss in detail. Novel genomic or therapeutic association, new indications for established drugs. Whatever drug which has been established, earlier we had not studied. If it is nephroprotective, you will say the new indication of this drug is nephroprotective. So the new indication becomes nephroprotective effect. Establishment of a new dosage form, I told you. Dosage form, you can change the dosage form and see the effect. And after seeing the effect, it can be established after doing on a large sample size. Pharmacogenomics and pharmacogenetics. Again, the gene variant, how the medicine is acting in a particular individual, this type of gene setup, if there is some genetic makeup, this genetic makeup in a particular individual, does it affect the effect of the efficacy of the drug what you have selected? Is it changing with the genetic type of the individual? That you have to study. Next. the bioactive molecules. What is the bioavailability? What are the active ingredients in the particular drug we have selected? And what do you mean by bioavailability? For example, the active ingredients of a particular drug, if you have taken what route of administration and how much bioavailable it was. For example, 100% of bioavailability is seen if you give the drug in intravenous route. So intravenous administration, directly it goes. If you push an injection, directly it goes with the systemic circulation and 100% bioavailability is seen. Therefore, you should know all this. What are the drug interaction? What are the side effects? Even that has to be mentioned. If there were some side effects, you have to definitely mention. What is a combination therapy? Have you given a combined drug? Next. Bioinformatics. So any information, whether it is a amino acid sequence of a drug, the DNA structure, everything will be fed with the help of the computer. That is using of the technology to save all the information. That becomes bioinformatics. Now the effective vehicle used to dissolve the active constituent of the drug. Sometimes if you are able to dissolve the active ingredient of the drug in water, you will write aqueous extract. If you require ethanol, then you will write ethanolic extract. So what is the vehicle in which you are able to dissolve your active constituent of the drug? That vehicle should also be mentioned. The nutraceuticals. So these nowadays, there is a scope of nutraceuticals for anti-aging. So for nutraceuticals for geriatric care. Now they have developed this kind of good superfoods. It is called superfood, wherein it will improve the health of an individual. In a person who is more than 50. So it is kind of preventing. Now what cosmetic procedures, there is a collaboration now going on where cosmetic procedures are being employed to look more younger. They want to, what the actual age is, they want to look 10 years younger, 20 years younger. They go for the cosmetic procedures. Now there is a collaborative research wherein cosmetics, any procedures, they give nutraceuticals. Why? Apart from the procedures, any invasive or non-invasive, they are giving some nutraceuticals so that it has a good impact and effect with this combination therapy. Next. So bioequivalent studies. So bioequivalent studies, again, it will be a comparison between a same generic drug, wherein you will see with the different dosage forms how it is acting, or with the different route of administration. One we are giving IM, when we are giving IV. So which is more effective? That is bioequivalent studies. Then pharmacoeconomic. With this, we are trying to find out a cost effective and something which benefits to the public. This is pharmacoeconomic. Then pharmacoepidemiology. Both. So therefore, we are trying to find out where the target actually is and how it can affect. The tissue regeneration. With utilization of certain drugs, the tissue gets regenerated. Next. So standardization and validation of an article for the drug research. See, while drafting drug-based research article, you should focus on standardization of the herbal drug. It refers to confirmation of its identity and determination of its quality, purity, detection of the nature of adulterant by various parameters like morphological, microscopical, physical, chemical and biological observations. So first thing is identification. After identification, for example, whichever institute you are sending, you have to just keep that reference number that you have identified it. After identifying whether this drug what you have selected, it has been standardized. So there are so many classical books. After taking the reference, standardization has been done by the Central Council Research Institutes. So validation of herbal drug requires pharmacogenetic, same thing. The taxonomy, the classification, whether it contains heavy metals, everything about the standardization has to be mentioned of a particular drug what you have selected. Now drug, same thing. The drug identification and authentication, I told you what part of the drug you have selected for the trial, whether it is a fruit, whether it is a flower, whether it is a bark, whether it is in the form of powder, flower, fruit, whatever you have selected, you are going to mention, you are going to send the sample to a particular institute and they will identify, they will authenticate, they will check the purity and give the letter. Next. The safety and efficacy of the drugs. This branch deals with the application of pharmacological principles in clinical settings. Drug-based research articles should include studying the effects of drugs on humans, including their therapeutic uses, side effects, interactions with other drugs. So, safety and efficacy. For efficacy, we'll see based on some how they have recovered, what were the signs and symptoms earlier, did they reduce, how the effect was, that is called as efficacy. For the safety we'll check the LFT, again renal function test, liver function test before and after the trial. Three broad categories of experimental studies. One is in vitro, that is inside a, like maybe the in vitro is in a petri dish or a test tube, in a controlled environment outside of a living organism. If it is inside the living organism, it becomes in vivo. And if it is just you are seeing a biological experiment carried out entirely on a computer or just a computer simulation to see what it is, that it is called as in silico. Next, same thing. So the Global Genomic or Therapeutic Association, the article of drug research, it deals with the interactions between the drugs, the disease and the genes and provides strong support for drug discovery and development. So now it has gone to such an extent, what is the action on the genes, the genomics. We are studying the genetic variant of a particular individual. If it is in say in few patients, it is having a better effect. So what was their genetic makeup? What was their genetic variant? Wherein this particular drug is acting. Why? Because at that level they are planning to treat, especially cancer patients. So the medicinal herbs or herbal formulae. What are the herbal ingredients? What is the then the drug availability? Then herbal constituent data. The drug target. What is the drug target? Then how the gene targets, what was the transformation and what finally happened, what is the result. Next. So establishment of a new dosage form. Article of a drug research. It is the need of an hour that herbal products must be formulated in various dosage forms such as decoctions, herbal powders, alcoholic beverages, capsules, tablets, ointments and creams. Hence, seeing this potential in the Ayush, they have opened so many centers of excellence. Or we can have a collaboration of centers of excellence. For example, in Bangalore, Indian Institute of Science, they have opened the avenues for Ayush faculty members, wherein they actually planned for a collaborative research. Same thing in Savitribai Phule University, there is an collaboration and that is called as Ayush Center of Excellence, wherein they are planning for the, since there is saturation, in allopathy side there is a saturation, whatever they have done, now they want to explore with the help of Ayush faculty members. So only the NCISM has taken an initiative to make you, aware of what all is going on, to see what the article and what collaborative research is. Next. The pharmacogenomics and pharmacogenetics. This emerging field combines pharmacology and genomics to study how genetic variants influence drug responses in individuals. Pharmacogenetics is the study of genetic causes of individual variations in drug response, whereas pharmacogenomics deals with the simultaneous impact of multiple mutations in the genome that may determine the patient's response to the drug therapy. So at that level, how they act, what is the genetic makeup and how we can target these cell lines, that level the research is going on. And even they are trying to make a patch of myocardium. For example, if there is myocardial infarction, there will be death, ischemia and death of the ejection fraction reduces. So now the research is going on to make a patch of myocardium so that after doing some operation they can fix the patch, they can increase the ejection fraction. So to that level the research is going on. At present research is being carried out for myocardial patches in Indian Institute of Science, Bangalore. It proves the concept wrong that same drug and dose fits all. So it proves the concept wrong. The same drug, the same dose fits all. No. When you go for the genetic variant, the dose can be altered, or it can fit, or it can be, it cannot work in some individuals. So next. So bioinformatics article of the drug research. Bioinformatics as related to genetics and genomics is a scientific subdiscipline that involves using computer knowledge or computer technology to collect, store, analyze and disseminate the biological data and information. I told you such as DNA, the amino acid sequences, everything will be collected and stored. And that is why in the paper where especially the drug research, they will write down all these sequences and what extract, what is the vehicle and how it acts, what are the properties, what are the active ingredients which are present, whether carotenoids, whether it is exhibiting antioxidant property. If it is exhibiting antioxidant property, what were the active ingredients present so that you can label it as antioxidant property. Carotenoids, flavonoids, if they are present, it is called as antioxidant property. So these properties, whether they were able to exhibit in the aqueous extract or whether it was some ethanol, any alcoholic extract, to that level you can find out and you can tell, yes, this particular drug has so many pharmacological properties. It has antioxidant effect, it has anti-inflammatory effect, it has then antibacterial, antiviral, whatever. What properties you have studied in which vehicle, that will be mentioned. So bioinformatics is a tool that helps researchers figure out the human genome, look at the global picture of a biological system, develop new biotechnologies. It will be used to create the personalized medicine of the future. So as per the individual needs, it becomes a personalized care in future. Every individual, it may change, the treatment may change, the pattern may change. Next. Selection of the research project. Article of the drug research. See, emphasis should be given to the drug, its development, standardization, safety and efficacy exploration, novel dosing, novel therapeutic efficacy, more economic and effective drug research. Before selecting any particular drug, all this has to be kept in mind. Easily available, cost effective, already standardization done. That kind of a drug you have to select and then go for the trial. A research work should be selected aiming broader societal benefits. So it should have more benefits on the society. The availability of relevant funding sources as well as resources for smooth completion. Simply you are going to collect a drug which is not easily available. Then how will you complete the trial which is not easily available? It has to come from Iran, it has to come from America. Such kind of a drug we are not going to select. So what is the availability? Whether it is easily available. Are the people ready to fund for it? Are we having available resources for the smooth completion? All has to be borne in mind. So whether it is, yes, easily available, the economical, whether it is cost effective, must be preferentially selected. The time limit for completion of the project should be drawn. It is very, very important. You will write within 1 year, within 2 years, I am going to complete this particular project. So the time limit is very important and you have to complete within the time framework. So selection of the research project. The author should ensure that procedures have been approved by the appropriate institutional ethical committee. So always before starting any trial, you should have an approval from the institutional ethical committee. It will be a committee framed by the institution wherein we will have odd number of members with one member secretary from the college and rest of the members, maybe one religious scholar, one layman, one lawyer, compulsory. one biomedical scientist, and one research. For example, anyone who has done on Adriya, that is Dravya Guna. If you are carrying out this drug research, that particular who has done PhD, PG, and who has good experience about research, he should be the member of that particular institutional ethical committee. Then only after their approval, we can start the trial. It must be registered with the Clinical Trial Registry, CTRI India. Now it's made compulsory to register. All are prospective. It is not retrospective. All the earlier trials, they had given a time limit to register the trial, though it was retrospective. But I think from past 5 years, 5 to 6 years, it should be prospective trial. They have to register. And author should also include a statement in the manuscript that informed consent was obtained from experimentation with human subjects. A complete information sheet in their own language has to be given. Has to be, in case if they are not able to read, explain them in their own language after showing the information sheet in English, in Kannada, or read out the information sheet, then take the consent. The privacy rights of human subjects must always be observed. After utilization of this particular drug, there is some good effect, but you have to maintain the confidentiality of the patient as well as take the consent from the patient that you are going to publish. In future, maybe for publication, you will use the data, but you will not reveal his identity. By maintaining his confidentiality, you will require the consent from him to publish the paper. It's always important to take this consent. Next. So again, the title. Again, you will be, the other guest speakers will be talking on the title. They will be talking on the introduction. There will be one whole discussion, 1 hour session on discussion. Again, if you want me to tell, I will tell you about the title. It should be precise, it should be more attractive, it should have the intervention, the study design, the disease. For example, here, what drug, what disease, you have to mention. Next, abstract also. You have to, always the abstract should be structured. If it is in the INRAD format, then it is called as structured abstract. What do you mean the structured introduction? Some brief introduction about the topic. Then you will write methodology. What is the methodology? How are you going to do it? What is your methodology? Everything you have to mention. Results. What were your results? Finally, after the completion of everything, this abstract will be written. So what were your results? What is your discussion, interpretation, and conclusion? If you write in this way, it is called as structured abstract. Next, introduction. See, though it is the first part in the thesis, it has to be written at last, introduction. Because you will actually know what has to be written after writing everything, a brief introduction, such that if you read the introduction, the reader should feel, yes, I have to go in detail. If the introduction gives some preliminary insights, some few insights which has been discussed later, then the person feels like reading your entire paper or the thesis. So this is introduction. Again, they will tell you what is introduction, everything in the coming lectures. Next, material and methods. One separate again. How do you go for material methods? How do you select? What is here the material and methods slightly differs from the clinical trial. See, material collection and authentication. How have you collected? What is the resource? From where did you procure it? Even that has to be written. After procuring it, did you get it checked? Is it authenticated? That also has to be written. What is the name of the drug? What is the botanical name? What is the common name? Which part you are using? Whether it is leaf, stem, bark, root, fruit. Which is the method of collection? Which season? Then what are the macroscopic and microscopic features? The chemical analysis, the pharmaceutical research. I told you, what are the pharmacological studies which will exhibit their properties? What are the properties? For example, what effects has been mentioned in the classical literature and the modern. If they have published on a particular curcumin, for example, curcumin, it is anti-inflammatory. Even in our literatures, they say that it is anti-inflammatory. So that anti-inflammatory you have to correlate. Yes, the modern pharmacological activities, proved pharmacological activities state that it has anti-inflammatory effect, and our classical books also say that it has anti-inflammatory effect. So this has to be, correlated. Then the dose selection, specification, what are the instruments used for standardization, what instruments you have used, what apparatus you have used, even that has to be mentioned. Then material and methods continued. Method of preparation, how did you prepare? Single poly, whether it is a single drug or whether it is a polyherbal preparation. Is it just the juice, whether it is kashaya, whether it is joshanda, Taisanda, joshanda, whether it is safu, that is a powder, churna, whatever you have to write down. Whether it is just an extract, herbo-mineral preparation, how did you prepare, whether you have used some mineral form, or you are going for any new dosage forms, what was written in your classical books, you have already conducted the trial, and what is the new dosage form, even that has to be mentioned. Then the route of administration, the doses and duration, how many times you are going to give the dose, whether it is TID, whether it is once daily, whether it is kashaya, given before food, everything in detail. What is the route of administration, what is the vehicle, sometimes we use the decoction. So what type of earth you are using, as Badripali said, the vehicle. So what you are using, the same thing has to be mentioned. The result and data presentation of drug research. Then finally, you are going to compile the observations of the study in the prescribed format. Usually it will be tabulated, bar diagrams, charts, figures, with footnotes at the end of each table. Graphical presentations, I told you, pinpointed description of the data, concise presentation in tables. Again, the biostatistician will help you to calculate the results. Yes. Discussion. Again, I told you a complete 1 hour will be given on the discussion. How to write discussion, but still I tell you, previous papers, you have what are your findings? What is given in the classical literature? Cross reference with the any other modern papers. So discussion. Again, it will be dealt in detail. I just don't want to go in detail about the discussion. 1 hour you will have only on discussion. Next. So in the discussion, I'll just summarize. In the discussion, you have to introduce the study. What is your study? What is the background? Summarize the main findings. Then tell about your results. Link back to the introduction, what you wanted. Highlight the implications for future research. Always, what are the recommendations? Address potential and existing limitations. What did you find in your study? What are your interpretations? And what would you recommend for the future? What is the limitation of your study? All this comes under discussion. And always you have to take the examples from the previous studies. These are the references. Thank you.
Thank you so much, madam, for your wonderful session. Now I request to assistant principal sir and the health secretary Dr. Ayush. Ma'am, please occupy the seat.
Madam, one file is first thing we have to do that while taking the sample, when we are comparing with the control and the grinder, the sample we can vary for the control. So when we are interpreting the results or statistical data, then how can we substantiate it?
Sir,
It will vary, no?
yes, it will vary. Already it is proved drug, no? Standard is a proved drug. For example, in a clinical trial only, for example, when you are taking a control, it is proved drug. We want to prove the efficacy of our drug. When you are taking treatment, group and or standard control. If your control is a standard control or a historical control. What do you mean by historical control? Historical control, your previous study or your colleagues have done some study. That particular it has not been approved from the FDA, but you have already proved that it has some better effects. So that can be taken as a historical control. Okay, sir. If you are taking a standard control which has already been approved by the FDA, why do you want to take the same sample size and probe? Right? What we will do is the biostatistician, we have to tell our intention. This is what I do when I prepare the synopsis protocol. I tell the biostatistician, when I'm taking a standard control, first of all, sometimes I don't like the standard control. I say, you do the comparative trial taking two drugs. Okay? When we are taking standard control, I tell them, give the ratio as 2:1. Where test group, the number will be more. Difference between the groups you are taking, if the number varies, it will be difficult. No, sir. When we tell them, they will interpret based on the 2:1 ratio only. Clear? So always it is better. What my thinking is, we will take into 2:1 or 3:1 ratio. For already the standard drug which has been already proved its efficacy, why simply we should waste our resources on the standard? If you take placebo, they will not agree. They will not agree for the trial. The ethical will not have the approval. So instead of standard, instead of placebo, we have to take the standard. For placebo, they will not agree. Therefore, we will take standard but with little minimal sample size. For our test group, we take more sample. Right? First of all, in the limitations, always you see in the limitations, in our trials, always the sample size will be less. Our sample size will be less. So at least, and for publications, when you go for the publications, if you don't have good number of sample size, then they will not publish your paper. So to avoid all that, but again ethical approval is also needed, we take the standard instead of placebo, or go for directly comparative between two drugs, or go for the same drug, different dosage. For example, I have selected one drug, alum, potash alum, in my recent trial. One will give intravaginally for heavy menstrual bleeding, and then will give per rectal. Per rectal misoprostol is given, but it has not been FDA approved. But it is the first line treatment in PPH, postprandial, PPH, postpartum hemorrhage. In postpartum hemorrhage, misoprostol is given, though it is not FDA approved. It is widely, followed practically. In the practice bulletin also they mention misoprostol. But the same misoprostol, when I take for the research, there will be ethical issues. Why? It has not been FDA approved. I can be land up in a suit. I will have to face the legal issues. So misoprostol cannot be given per rectal because it is not FDA approved. So what I thought, I can go for two different dosage forms. One intravaginal and one per rectal. Because nowadays intravaginal route and also per rectal, they are doing extensive research on changing the routes of administration. So and in our classical literature, so long back they have given us intravaginal and per rectal. So why can't we use that? So I got the ethical clearance for per vaginal also, per rectum also, but I didn't get the approval for misoprostol. So always we should also think. But it will come with experience. And always for a standard control, take a less sample size. For your test, take a good sample size. Okay? 2:1 or 3:1. Thank you.
statistician can decide.
Yes, you should have a good rapport with the statistician and also sit and discuss what you want. The statistician, based on the scale, the score, or the previous study, they will help you. They'll guide you.
Thank you. Madam, for animal experimentation, specifying the age is mandatory.
Specifying the age is mandatory, sir. Why? Because, for example, we are studying about the cardiovascular diseases, and cardiovascular diseases usually it start late onset, right? In 2-3 months it will not initiate. That changes. So 2 years. In the 2 years model, so we selected if there is some cardiovascular changes seen in a particular rat, age 2 years. So depending upon the disease, you have to tell about the age, and you will have so many papers. For inclusion criteria and exclusion criteria also, you should give the reference. Some reference. For example, any paper which has been already published, they have seen the effect. But this particular drug, what you have selected, they have not seen. But in case of inclusion criteria for cardiovascular diseases, 2 years of age, you can give the reference. It has been published as per the RI guidelines. You just give the reference and take the age limit. So age limit is important. You have to mention. Yes, sir.
As in the human being research, so we are doing this sample size calculation.
Yes.
Similarly, in case of animal experimentation, is there any formula or anything criteria for doing the calculation of sample size for animal experimentation?
Yes, sir. For that also you have sample size estimation based on previous study only. Based on any scale. For example, I told you pain scale, right? Glasgow Coma pain scale. So that pain scale only, the pain scale, depending on the pain scale, what were the variables, how it reduced, what percentage it was reduced. Based on that, always it is better to go to a biostatistician and tell based on what is the sample size. For example, if it is a prevalence trial, if you want to go for the prevalence, there will be huge number of sample size. And you are doing in a controlled environment, you should tell them first, you want this amount of sample size. So they will take 2 or 3 papers, 2 or 3 scales and try to come to your sample size. So you also should tell them what sample size you want. For example, prevalence, if you are doing the trial, in a human trial you are talking about, prevalence trial 400, 500, 600, sample size. So prevalence. Based on earlier prevalence papers, they will calculate. If you want to go for a clinical trial, if you want to go for a randomized control, that is 2 groups. If you want to go for pre and post analysis, 1 group. If it is 1 group, you can go for n is equal to 40. 40 sample size is enough. Or at least 30 should be the minimum sample size. If you are going for 2 groups, there will be some 10% dropout. Dropout is also common with 10%, with 20%. Again, based on that 10% dropout also you should keep in mind. And then you should calculate the sample size. If there is any difficulty, you please take the help of the biostatistician, and any PG institute should have a biostatistician. So with that, what type of trial you want to go ahead, you have to tell your biostatistician. If it is single group, you want the sample size as 30 to 40. If it is a randomized control trial, 2 groups, let's say madam minimum of 30 sample size. So with the dropout they make it 30, without dropout they make it as 28, 27. So with or without dropout, actual sample size should be written in methodology what you write. Actual sample size n is equal to 27 in each group. Okay, sir. Then you will write with the 10% dropout, this becomes 30. It becomes 28. So in that way, if you tell to your biostatistician, they'll help you. For animals also same. Yes.
Madam, the form of medicine and route of administration is different. Whether comparative study can be done?
Sir, actually the form of medicine should be same. When you are doing comparative study, for example, we are preparing suppository. You are changing the routes, but the form should be same. For example, I told you earlier the example. We are preparing a suppository with potash iron. So the preparation, the dose, everything will be same. Only the route will change. Otherwise, if you want to go for the change of the dosage form, the route should be same. The group should differ. This way.
Madam, the question. Regarding animal vaccine experimentation, already the European Union has banned animal experimentation.
Yes, sir.
And the beta, so forcing government of India to ban animal experimentation. What is your opinion regarding this?
Sir, if you ban the animal experiments, will it benefit the humans?
They are finding some alternatives.
Yeah.
Part of this.
Mhm.
And according to US FDA, the accuracy of animal experiment is only 8%.
Yes, sir, that's true. I don't know, 50% of the research going where
What
just.
is the authenticity of this animal experimentation?
Sir, actually, though they are banned, but they are spending dollars on animal research. You just open and see the US how many billions of dollars they are spending on the animal research. Can you think preparing a myocardial patch, is it possible without animals?
No, in some part it may help. I'm not telling you 8% is there.
I don't think so they should ban. I don't think so they should ban.
Well, already it has been banned in European Union.
But India is going on and they shouldn't ban.
No, in toxicity study it may help.
Yes.
Some of the things, but accuracy, when you see the accuracy, it is very least. So they are finding some alternatives. And another question is you talked of different formulations in herbal medicines. Herbal medicines are used in imaging and all those, and all those formulations which you talked regarding decoctions, alcoholic preparations, and all those. They are there, and thousands of research works have been done.
Yes.
Already they have been published.
Yes, sir.
And even the pharmacy people are running on these herbal medicines, especially the formulations, where they have also published it scientifically.
Yes, sir.
So I think that point is, when you go for research should be done. Already it has been done.
Oh yeah, yes sir, I agree. I
We
totally
have all
agree.
those formulations.
We have, but just for cross-reference, we have to say for the cross-reference.
No, I don't have any doubt, but the curiosity, in your first presentation, there was a picture with 3 hands. So what that means? Please show that picture.
Integrity, sir.
But the hand had 6 fingers.
Oh, I didn't notice. Oh, the hand has 6 fingers. Let me see. Yeah, good observation. Okay. Anything else, sir?
No, it was said that the PPT were prepared and scrutinized and finalized. So it might have some implication or importance, so that is why I asked.
No, sir. I didn't notice that.
Please show this slide. I may be wrong.
Six fingers.
extra growth area. Identification. This for identification.
Short break, tea break, and the next session will start immediately after 10 minutes.
Thank you all.
So it is an informative session. So shall I get your number? Then you can photographer.
ಪಾಸ್ ಮಾಡು ಏನು